these are notes that are going to go into your HBS course file. These are notes on the muscular system. And so once again, I have a post-it that I put at the top for the muscular system so I can get right to that page. So here's my little muscular system post-it so that I can get right to the page. And on the back of the post-it, I give myself the functions of the muscular system. Functions of the muscular system, very simply, is to allow movement through contraction and relaxation of muscle fibers. So that's basically what muscle fibers do, is they contract and relax. And that contraction relaxation allows for movement, no matter what type of muscle we're talking about. All right, so first half, top half of the page, maybe about a third, top third of the page, I have the different types of muscles. And so we kind of looked at this with our tissue insert foldable. We have voluntary and involuntary muscle. So voluntary muscle is our skeletal muscle. We have voluntary control over this muscle. Involuntary muscle we do not have control over. So examples are cardiac muscle. Cardiac is specifically for the heart. And smooth muscle, so intestines, the diaphragm, lots of our digestive system, organs that have muscle, blood vessels, the muscles with the arteries, we don't have any control over those as well. They are involuntary. So for involuntary, there's no conscious control or limited conscious control. So you can hold your breath for a certain amount of time, and so you are exerting a certain amount of control over your diaphragm, but you can't do it forever. All right, so striated muscle is muscle that looks like it has stripes. The striations in striated muscle are created by actin and myosin fibers arranged in sarcomeres. So striated muscle is muscle that looks like it has stripes. The striations are created by actin and myosin fibers arranged in sarcomeres. Cardiac muscle and skeletal muscle are striated. So it doesn't matter whether it's voluntary or involuntary. Voluntary means that you can control it. Involuntary means that you can't. Striated has to do with the organization of the actin and myosin fibers in the muscle. So cardiac muscle is striated muscle. Skeletal muscle is striated muscle. Smooth muscle has actin and myosin fibers, but those fibers are not organized into sarcomeres, and so it does not have that striated configuration. Smooth muscle has myosin and actin, but the fibers are not organized into sarcomeres, and so it is not striated. All right, so a little review. Tendons attach muscle to bone. Ligaments attach bone to bone. All right, next. So that was these notes right here. On the next page, you will end up with another graphic organizer that you do a muscle build, and so you're going to draw stuff on there. So we'll just take that in. The next page, we're going to talk about the different parts of a muscle. So on this page, parts of a muscle that helps me to find what I'm looking for in a review or if I'm answering questions. So I've got my diagram up here at the top. So this is a tendon because it is attaching muscle to bone. We're going to go through these other terms. Epimysium, that's talking about this layer right here. Fascicle is talking about this level of organization. This is the muscle. This is a bone. Blood vessels, because we have blood vessels throughout. Perimysium is this lining. Endomysium is this lining. And then this would represent a myofibril or a muscle fiber. So a myofibril is the thread that a myocyte is composed of. So myocyte, myomuscle cyte cell. So myofibril, the threads that the myocytes or the muscle fibers are composed of. Endomysium is the membrane that's around each muscle fiber. So you have a protective membrane around the muscle fibers. The fascicle is where we take those myofibrils and we pack each myofibril into a bundle. So each myofibril has an endomysium that it's encased in, that it's protected in. And then all of those units bundled together form a fascicle. The paramecium is the membrane around the fascicle. So you have that bundle of muscle fibers, and that bundle is a fascicle, and then the fascicle has a membrane around it as well. The epimysium, the epimysium is a tough outer membrane around a group of fascicles to form a skeletal muscle. So the fascicle is going to end past the muscle fibers and come together to form the tendons and attach the muscle to the bone. So the epimysium essentially covers the muscle, and then where the muscle ends, it becomes the tendon, and that attaches to the bone. So the epimysium, the tough outer membrane around a group of fascicles to form skeletal muscle. It ends past the muscle fibers and comes together to form tendons and attach muscle to bone. So this is taking a muscle fiber and kind of looking at it in a much more closer look. So we're magnifying this. Has a higher magnification here. So we have the sarcolemma, sarcoplasm, sarcoplasmic reticulum is the blue part right here. The sarcolemma is a cell membrane specifically for the muscle fiber. So sarcolemma is a cell membrane. All cells have cell membrane, but it's called a sarcolemma when we're talking about a muscle fiber. Sarcoplasm is cytoplasm. All cells have cytoplasm. When it's talking about a muscle cell, it's called sarcoplasm. Cytoplasm of striated muscle cells. Sarcoplasmic reticulum, it's like endoplasmic reticulum, just endoplasmic reticulum in the muscle cell. That membrane specifically in the muscle cell is specific to store calcium because that's going to be important in our muscle contraction. So sarcoplasmic reticulum is a transport membrane. It's just like endoplasmic reticulum, but in the muscle cell it stores calcium. So sarcoplasmic reticulum is the blue part right here. This is our little muscle cell. Sarcoplasm is the cytoplasm in between all of this. Sarcolemma is the cell membrane on the outside of it. All right, on the next 2 pages, the sliding filament theory. The sliding filament theory. So all of this has to do with a muscle contraction. It has to do with the sliding filament theory. Sliding filament theory is called a muscle contraction. So I put this here so I'm not looking at parts of a muscle, I'm looking at how the muscle works here. So that gives me a better idea of what I'm looking at. All right. I guess I'll do this one first. All right, so here's a diagram that kind of summarizes and simplifies what we've got going on here. Number 1, ATP is hydrolyzed when myosin head is unattached. No, let's go back to looking at these myosin and all these filaments. Okay, so I'm going to do this one first. All right, so a muscle fiber can contract a maximum of 50%. That's it. The sarcomere is the segment of the myofibril that contracts. So the sarcomere is what is contracting. Here's our sarcomere, here's our unit, and this is what's going to contract. So this is one sarcomere, this is another sarcomere, so this is going to contract, this is going to contract. The myofilaments that make up a sarcomere, those myofilaments are called actin and myosin. So actin is a thin protein filament. It's anchored to the lateral sides of the sarcomere. The Z line, it kind of looks like a Z. I'll show you in just a second. The Z line is the actin myofilaments. Myosin is the thick protein filament, and it's anchored to the medial portion of the sarcomere, to the middle. So it's going to pull actin into the middle during muscle fiber contraction. So our muscle fibers within the sarcomere, here's our sarcomere. See the Z line? That's going to be our actin. So the kind of purpley bluish one, that's our actin. Myosin, it's in the middle. So it's anchored in the middle, and it's going to pull these actin fibers, it's going to pull those actin fibers, it's going to pull them together so that they contract. Underneath here, that's kind of taking what we just looked at. Here's our actin fibers, our little Z line. Here's our myosin anchored in the middle, and it's going to pull that in toward the middle. So actin, our thin filament, I highlighted that the same color. Myosin, our thick filament, I highlighted that the same color. All right, a couple of things that are important, a couple other components of those muscle fibers that are important in our sliding filament theory. Tropomyosin and troponin. Tropomyosin is a protein fiber that runs the length of an actin filament, blocking the myosin and binding and blocking the myosin binding site on the actin. I can't even read my own handwriting here. Tropomyosin, protein fiber that runs the length of an actin filament, blocking the myosin binding site on the actin. So this is kind of like a thin thread, if you will, that goes down the actin filament, and there are sites that allow troponin, allow those myosin heads to bind to it. And if it's blocked, the myosin heads can't bind to it, and if it can't bind to it, it can't pull it in toward the center. Troponin, calcium binds to troponin, and these are like little sites. These little sites change the troponin shape, causing that tropomyosin to get moved off the binding sites. So tropomyosin is kind of like a thread, if you will, that goes entire length of the actin. The troponin, it has sites where calcium can bind to the troponin. It changes that troponin shape, and it causes the tropomyosin to move off the binding sites on the actin. All right, now this might make a little bit more sense here. So, myosin head, our actin filament. On the actin filament, that thread that kind of goes along the whole actin filament and covers up these little binding sites, that's our tropomyosin. The troponin is this little binding site where calcium can hook onto. And so when calcium hooks onto the troponin, it changes its shape and exposes these little spots, the little myosin binding sites. So first of all, ATP is hydrolyzed. The myosin head is unattached. That causes ATP to be hydrolyzed. So this is the myosin filament. This is the little myosin head. Myosin unattached. ATP is hydrolyzed. Remember, hydrolysis means the breakdown of a polymer. So ATP being hydrolyzed means ATP is being broken down. When ATP is broken down, it's changed into ADP and a phosphate group. So ATP is hydrolyzed, so we have ADP and a phosphate group on that myosin head because the myosin head is unattached. Next, ADP and a phosphate group are bound to the myosin as the myosin head attaches to the actin. So it's attaching to one of those attachment sites, binding sites. Next, ADP and the phosphate group release, causing the head to change position and the actin filament to move. So before, it just bonded. Now when the ADP and phosphate group are released, it creates the power stroke. It creates the movement that's going to pull that actin fiber in toward the middle. ATP, because that ADP and phosphate group go to the mitochondria and are reformed into an ATP molecule. Then that ATP molecule that reforms binds to the myosin head and causes it to detach and return to its resting position. Then it's hydrolyzed, it's broken apart, that allows it to bond. It releases, causing the power stroke, gets reformed back into ATP. When that bonds to it, it goes back into its resting position. But this has already been pulled, and so this pulls it and then resets, and then it can pull it again and then reset and pull it again and then reset, and that's what allows it to continue to moving closer to the middle. Sliding filament theory. Okay, yeah, we'll do that in class. Sliding filament theory, muscle contraction. That's what we got going here.